We talked about last class aspects related to phosphocreatine degradation, so basically the anaerobic or non-oxidative ATP supply systems. We're going to move a little bit now into oxidative phosphorylation, so aerobic ATP supply systems, some of their characteristics, roles, and limitations. And from there, move into some of the factors related to the control of bioenergetics, VO2 uptake kinetics, and how ultimately these three energy supply systems communicate with one another. And why oxidative phosphorylation, which is this inexhaustible supply of ATP that can maintain energy and cellular homeostasis and balance and limit the development of fatigue and so on and so forth, doesn't turn on automatically at the onset of exercise, and why there is this interdependence between the aerobic and anaerobic energy supply pathways. So this is the mass balance equation for oxidative phosphorylation. To the left you have the substrates, to the right you have the products. The most critical product, of course, is the production of ATP, which drives energy and muscular contraction. So in terms of the substrates, we have the NADH that is produced through glycolysis, so breakdown of foodstuffs. Glycolysis produces NADH, which can be shuttled into the mitochondria, as we talked about last class. And then also, as you'll see, NADH as a form of potential energy is produced in the Krebs cycle through the breakdown of foodstuffs, proteins, fats, and carbohydrates. We also have oxygen as a critical substrate in oxidative phosphorylation, because it is ultimately the final electron acceptor. And so as you'll see, it helps to maintain the electrochemical gradient, hydrogen-ion gradient of the electron transport chain, and allows oxidative phosphorylation basically to continue. So if there's enough oxygen, saturating levels of oxygen, oxidative phosphorylation will move forward. We have hydrogen ion, ADP, and inorganic phosphate. So we have the substrates of oxidative phosphorylation, which in the presence of mitochondrial enzymes, so again, whether or not we have a lot of enzymes regardless of their activity, or the activity of those enzymes regardless of their concentration, we kind of have this boosting effect, right? The enzymes catalyze reactions to make them move more quickly. So for any given amount of substrate, we can move through this oxidative phosphorylation mass balance equation if we have more active or more enzymes. And then, of course, in terms of the products, we generate ATP and water and NAD as a byproduct of oxidative phosphorylation. And so the characteristics of oxidative phosphorylation or aerobic ATP production are effectively that it's an inexhaustible substrate supply of ATP. So we're not limited by the amount of stored ATP, we're not limited by the amount of phosphocreatine, as an example. So you can keep regenerating ATP inexhaustibly as long as you can continue to provide the substrates for it to occur. Okay, so it's kind of this inexhaustible, very reliable source of ATP production. Importantly, it's really that form of ATP production that will allow for stabilization of cellular homeostasis. So it's the form of ATP production that we would like to activate as quickly as possible and get to some steady state level as early as possible to prevent major metabolic disturbances into the interior, into the milieu of the cell. So again, as you'll see, one of the major things that we're trying to prevent in terms of disturbances are the levels of these particular metabolites, and most notably is inorganic phosphate. I'll show you later on that there's kind of a historical concept that lactate and hydrogen ion through the reduction in intracellular pH is really what causes fatigue. But actually, at a biological muscle temperature, is that pH really doesn't have much of an effect on tetanic force production. And so won't get into the details, but historically, I think it's one of those situations where the experimental procedures were not sophisticated enough to be able to look at the role of hydrogen ion or pH in muscle force production. Whereas as techniques became more sophisticated, people began to realize that in the right experimental condition, having a low pH or a lot of hydrogen ion really wasn't the most critical factor contributing to an impairment in a release of calcium from the sarcoplasmic reticulum. Hydrogen ion is believed to kind of compete for calcium on troponin binding sites, which compromises myofibril calcium sensitivity and so force production. But in fact, at temperature corrected kind of muscle temperatures during exercise, it seems like the contribution of hydrogen ion to muscle fatigue and force loss is actually pretty modest. Point is that you probably heard a lot about muscle acidosis and lactic acidosis being what causes fatigue. In fact, it seems to be that inorganic phosphate is the most important biochemical determinant of fatigue. And so trying to keep inorganic phosphate within narrow regulated levels seems to be very important in terms of preventing fatigue development. So again, you need to try to keep these metabolites in check and at low enough levels to allow muscle power output to be sustained. Again, what did we said that a performance limitation occurs when the muscle is not able to provide the power or the external force required for a particular task. Right? So we had those lines during incremental and constant load exercise where the demand of exercise was constant and the time at which you can perform that constant load of exercise is dictated by the rate of accumulation of fatigue causing metabolites in the muscle. So if inorganic phosphate is one of those very critical fatigue causing metabolites, you would like to prevent the rate at which it accumulates. One of the ways to do that is to try to turn on oxidative phosphorylation more quickly and rely less on phosphocreatine degradation and glycolysis as energy sources. Okay? So, the role of oxidative phosphorylation is that it uses the potential energy available from the reducing equivalents NADH and FADH formed in glycolysis in the Krebs cycle, ultimately to rephosphorylate ADP to ATP. So to recombine ADP and PI to create ATP, which in turn is used for muscular contraction. And it really provides prolonged ATP supply for continued power output activities. So things like marathons, long-term swimming, things that are of relatively less intense exercise but of longer duration. And the limitation of this system is that it's relatively slow to initiate ATP production. You'll see what we'll call is there's this metabolic inertia. So basically, I grew up in the late 70s, early 80s, and so we had this, anybody ever heard of a Cutlass? They don't even make Cutlass anymore. Anyway, my parents had this massive car. It was called a Cutlass Supreme. And it had a massive engine, two-door, and you couldn't get off the line quickly at all. But once the engine started rolling, it just drove beautifully. And I always think about this Cutlass Supreme in terms of oxidative phosphorylation, is that it can't get off the line very quickly, but once it's rolling, it's such a beautiful drive. Okay, so I kind of think of it like that, is that it's relatively slow to initiate ATP production, but it's nevertheless the car that you want to drive. And so, unfortunately, it's because of its sluggishness and the fact that it's relatively slow to produce ATP relative to stored ATP, phosphocreatine degradation, and glycolytic flux, which are much faster metabolic processes, it's unable to meet the ATP demands for very high power output activities. Things like, sprints or powerlifting or whatever it might be. Okay? So, again, I believe this is all stuff that you're quite familiar with, right? Yes, no? All right. I'll learn to read your guys' signals as we go along. So where does oxidative phosphorylation occur? Of course, it occurs in the mitochondria, the powerhouses of the cells, and involves the interaction of the Krebs cycle and the electron transport chain. The Krebs cycle produces in 3 stages basically the reducing equivalents or the energy to drive the electron transport chain. So Krebs cycle produces the NADH and the FADH, which provides the electrons to create the electrical chemical gradient of hydrogen ion to ultimately drive the oxidative complexes of the electron transport chain. So the Krebs cycle, again, it completes the oxidation or removal of hydrogen ions of foodstuffs, so proteins, carbohydrates, and fats. Of course, it produces CO2 as a byproduct. CO2, as we'll see later on, is a volatile acid. So CO2 can liberate, ultimately through a series of reactions, hydrogen ions systemically. So maybe at the level of the muscle, that's not particularly critically important, but in terms of, you know, what I was just saying in terms of developing fatigue, but if you held your breath right now, how long could you hold your breath, and why can't you suffocate to death? Why can't you suffocate yourself to death? Yeah.
Because your brain is going to override that control that molecule.
So that's kind of like where we get to, but what is the step, how do you get from CO2 to that brain overriding that control? What does the brain sense?
The brain senses the level of CO2 in the blood.
So it senses the levels of hydrogen ion, right, or the pH. So carbon dioxide will be retained through a series of steps. It will liberate a hydrogen ion. The hydrogen ion will cause acidosis. You've got chemical sensitive receptors in the carotid bodies that taste basically the acidity level of the blood going to the brain. And it also, in the brain, you've got central receptors that are very sensitive to pH as well. And so these reflex related sensors will become very, very active, and they'll drive ventilation. And so if you increase ventilation and excess of metabolism, you'll blow off CO2 as a means of trying to increase pH or cause an alkalosis to try to combat the acidosis. So of course, the Krebs cycle and the production of CO2 and its move into the blood can have a potential effect on causing acidosis and causing an acid-base disorder. Okay? The Krebs cycle supplies electrons in the form of the reducing agent, so NADH and FADH, again, as I mentioned, to pass through the electron transport chain to rephosphorylate ADP to reform ATP and to generate the energy for muscular contraction. And oxygen itself doesn't participate in the Krebs cycle, but it is the final electron acceptor at the end of the electron transport chain. So if there's not enough oxygen and moving the electron transport chain, basically you'll back up and the Krebs cycle won't operate effectively. So in this way, there is a relationship between oxygen and Krebs cycle activity. Okay? So, oxidative phosphorylation occurs in 3 phases. Quite simply, the first phase, very descriptively, is that there's a generation through the breakdown of protein, sugars, so glucose in the blood or glycogen in the muscle or in the tissue, as well as the breakdown of triglycerides through the process of lipolysis to ultimately produce acetyl-CoA, which is the precursor to enter into the Krebs cycle. The second process is the Krebs cycle itself, which is the oxidation or the generation of these reducing equivalents. So the electrons or the hydrogen ions to drive the electron transport chain through the Krebs cycle. And so we get this oxidation of acetyl-CoA generated through these foodstuffs in the Krebs cycle, which ultimately provides the energy to drive that electron transport chain. And third, we have, of course, the oxidative phosphorylation, which is the aerobic production of ATP in the ETC itself. So, forming acetyl-CoA from glycolysis, this is just a simplified version. Again, I blanked out all of the different Krebs cycle enzymes. I've only really noted 2 of particular importance because they're quite important in terms of rate-limiting enzymes. Everything else we talked about at the beginning of last class, we're not going to go into the details of all of the different enzymes, but we'll highlight just a couple. So we know from last time that pyruvate is broken down from last class into acetyl-CoA and to carbon dioxide catalyzed by a rate-limiting enzyme, pyruvate dehydrogenase. So again, if PDH activity is too low or if more pyruvate is being produced, they can be utilized and converted into acetyl-CoA. Pyruvate has 2 fates, right? It either converts into acetyl-CoA to be used by the Krebs cycle, or it's converted into lactic acid, which dissociates into the lactate and the hydrogen ion. And that hydrogen ion itself, again, can have a negative effect on intracellular as well as whole body systemic pH. As hydrogen ion levels fall, we become more acidic, and that has the potential to compromise muscle protein structure and function and contribute obviously to fatigue, but perhaps not to the extent that it was originally believed. Acetyl-CoA will then combine with this oxaloacetate to form citrate and enter in this tricarboxylic acid cycle or the citric acid cycle in a reaction ultimately catalyzed by the enzyme citrate synthase. So again, you see that we have a couple of potential rate-limiting enzymes and the activity of which are very critical to how quickly we can basically activate the oxidative phosphorylation. So how quickly acetyl-CoA can be produced through glycolysis, the activity of PDH, the activity of citrate synthase as a Krebs cycle enzyme, and then ultimately the activity of this isocitrate dehydrogenase, which appears to be the rate-limiting enzyme of the Krebs cycle itself. Okay, and so this is one of those enzymes that, as you'll see, are actually activated by changes in the energy state of the muscle, or the energy state of the cell, through contraction-dependent increases in calcium levels, inorganic phosphate, and ADP that occur through stored ATP breakdown, as well as phosphocreatine degradation and the rephosphorylation of ADP to ATP. Okay, so I'll show you that later on. Then there's a series of reactions that ultimately serve to regenerate this oxaloacetate, again to convert this acetyl-CoA and allow the Krebs cycle to continue, produce carbon dioxide. Ultimately, the ultimate goal of the Krebs cycle is to produce these reducing agents, or the energy to drive the electron transport chain, and to produce this GTP, or guanosine triphosphate, which itself can actually transfer its phosphate group to ATP to produce ATP itself in a process called substrate level phosphorylation. But basically, the Krebs cycle, the primary role, regardless of the nature and source of the foodstuff that's breaking down to the acetyl-CoA, is to provide energy for the electron transport chain. Okay? So we also have the formation of acetyl-CoA from fats or triglycerides, and this process basically occurs through lipolysis and beta oxidation. So fats that are stored in the muscle, but primarily in adipocytes or fat cells within the body, so these fats or triglycerides are broken down by enzymes. Again, there's various forms of lipases, so hormone-sensitive lipase, lipoprotein lipase, but there are these lipase enzymes, these fat-specific enzymes that will break down these triglycerides in the blood and different muscle tissues and different body tissues to ultimately form these free fatty acids. So these fatty acids will be produced. They'll move out into the blood, they'll combine with the albumin protein to produce this free fatty acid for transfer to different tissues and subsequent utilization. And these free fatty acids as well as glycerol. Okay? And so this ultimate process of breaking down the fats and triglycerides is a process known as lipolysis. Lipolysis increases blood and muscle levels of free fatty acids, and once we have this increase in free fatty acids, that actually facilitates fat metabolism. So basically, the rate at which lipolysis can be activated determines in part the extent to which we can have fatty acid metabolism. We know from the energy yield of glucose breakdown and fat breakdown that fat is a much better source and efficient source of ATP production. We get much more ATP from fat production, from fat as a fuel source, than we do from, say, sugar as a fuel source. But it's slower to generate that ATP. Okay. So free fatty acids then undergo beta oxidation to ultimately form this acetyl-CoA, which again will enter the Krebs cycle, generate the reducing equivalents to drive the electron transport chain. So the activity of these lipases, and thus the activity of lipolysis, does increase progressively during exercise. So exercise intensity dependent increases in the activity of these lipases and the enzymatic activity will increase the rate at which these free fatty acids are liberated and become available for utilization. Unfortunately, lipolysis is a relatively slow process, and fat metabolism occurs more slowly, and so fat metabolism is not an immediate source of energy at the onset of exercise, but fat metabolism is a great source for long duration type exercises like marathons, ultra swimming, various things like that. Right? So, the electron transport chain, basically as a as a brief review, it occurs in a series of steps. We have the cytosol, the outer membrane, the outer compartment of the mitochondria, the inner and the inner compartment here. And so the reducing agents, so NADH and FADH that were ultimately formed through glycolysis and the Krebs cycle, will transfer their stored energy in the form of these electrons to these complexes in the form to the electron transport chain, as you can see here. As these electrons ultimately move through these four cytochrome chains, my mouse is dead. Some of their energy is used to pump hydrogen ion from the outer compartment into the inner compartment. And so basically it's this energy through these electrons that creates this electrochemical hydrogen ion gradient between the inner and the outer compartment of the mitochondria. And so ultimately you get a higher concentration of hydrogen ions in the outer compartment than in the inner compartment. And this creates this electrical chemical gradient, which creates an energy creates a strong drive for hydrogen ion to diffuse back into the inner compartment, and is the potential source of energy used to, it got cut off, to rephosphorylate ADP and produce ATP at the level of this final assembly here. So ultimately, the movement of hydrogen ion through these respiratory assemblies of this inner membrane along the electrochemical gradient will activate the enzyme ATP synthase that you saw in last class, which catalyzes the phosphorylation of ADP to ATP. So ultimately, the electrons, the hydrogen ion, and oxygen at the end of this electron transport chain will combine to form water. So you can appreciate then that oxygen is considered the final electron acceptor in this electron transport chain. Oxygen, therefore, is also responsible for maintaining that electrical chemical hydrogen ion gradient, and therefore that energy gradient to drive this final complex and the rephosphorylation of ATP. And so thus oxygen allows for oxidative phosphorylation to continue. So if there's not enough oxygen, the rate of oxidative phosphorylation doesn't proceed at the same rate. And that can be predicted by that mass balance equation, right? The substrates to products. If you decrease one of the substrates without changing any of the other substrates and don't change the activity or number of enzymes, the rate at which you'll cycle through that mass balance equation will decrease. So one of the ways to compensate, if you have less oxygen, you could provide additional substrates through other pathways. So inorganic phosphate, ADP, and NADH. Those are provided through phosphocreatine degradation, stored ATP degradation, and glycolysis, right? So if there's not enough oxygen or oxidative phosphorylation is not moving quickly enough, the oxidative production or the oxidative phosphorylation can drive more quickly by providing other substrates other than oxygen. But that will require the activation of these anaerobic pathways, which again have fatigue implications because they produce or liberate things like, they result in the accumulation of things like hydrogen ion and particularly inorganic phosphate, which we'll see are negative in terms of muscle function. So ultimately, sorry that it got cut off, but you hear a lot about somebody's peak rate of oxygen consumption or their maximum VO2 as a measure of their aerobic working capacity or their maximum cardiorespiratory fitness. And the reason for that is because the rate at which oxygen ultimately is consumed by the body is an indication of how well our muscles are able to produce ATP aerobically. So the higher the maximum rate of VO2 that we can achieve, the higher rate of oxidative phosphorylation our muscles can participate in. So it's really the amount of oxygen that we can measure as being consumed at the mouth, which, for lack of a better explanation, you measure a percentage that goes into the body, you measure a percentage that gets blown out, take into consideration some time lags and various other factors, and you can get a rate of exchange. How much oxygen was consumed in the time from the breath in to the breath out, and you get a rate of consumption, right? So we know that we got 21% coming in. If we have progressively decreasing levels coming out, we have a greater rate of consumption. for a given unit of time, we can achieve these high levels of VO2, which is ultimately an indication of our ability to drive oxidative phosphorylation and support ATP provision aerobically. So with all of that in mind, we'll just kind of frame the theoretical concept of why measuring VO2, or the rate of oxygen consumption in a human during an exercise test, is an indication of somebody's aerobic working capacity. So consider this series of events. We're basically going to go from left to right, from top to bottom, and then it's not going to kind of follow a normal, follow the arrows is basically what I'm saying. So, what I just mentioned to you was that measuring the amount of oxygen consumed through measurements of gas exchange, which you'll do in the lab this week, Thursday and Friday, you will measure the rate of oxygen consumption measured at the mouth through this pulmonary gas exchange. Okay, so it's a non-invasive measurement of oxygen consumption. If you believe what I just said, then the rate of oxygen consumption increases in direct proportion to the amount of ATP produced through aerobic metabolism or oxidative phosphorylation. So, more ATP produced through oxidative phosphorylation, we get a higher VO2. So, VO2 is directly related to the rate at which ATP is produced via oxidative phosphorylation. So, how do we get to exercise testing from this? Well, we know that ATP supply must be matched to demand, right? We said that ATP is basically a regulated variable. And so, ultimately, we know that the rate of ATP supply needs to be directly proportional to the rate of ATP demand. Now, we've got three energy systems that are going to try to facilitate that matching of ATP supply to demand, right? So we have a need for 100 molecules of ATP. Not 100 of those molecules are going to come from phosphocreatine degradation, or from stored ATP, all from glycolysis, or all from oxidative phosphorylation. There's going to be a ratio, but we know that we have to match ATP supply to demand. Ideally, we would love to be able to make that match between supply and demand all through oxidative phosphorylation, but we know that that's not possible. We also know that there is a linear relationship between exercise intensity. So when you're on the cycle ergometer on Thursday or Friday, every 2 minutes, the external power output, or the resistance on the pedals, will go up by 25 watts. And you'll keep exercising until you can't push the pedals anymore, or until you decide that the symptoms are no longer tolerable and you want to stop. Okay? But there is a linear increase between power output, or exercise intensity, running up a hill. Right? If you're running at a constant speed and the hill continues to get steeper and steeper and steeper, the power output requirements go up and up and up. The ATP demands will go up in an exercise intensity dependent manner. So in this case, power output increases and ATP demands increase in direct proportion to those power output requirements. So the rate of ATP demand is then directly related to exercise intensity, what the muscles are required to do in order to support a given external task: walking, cycling, running, swimming, whatever it is. So, to come back to this, we can get two measurements that we can measure. Without biopsies and very sophisticated, magnetic resonance imaging studies, very, very tough to get a measurement of ATP supply and demand. But we can, through these series of assumptions, we can begin to look at the rate of oxygen consumption, which is measurable during an exercise test, relative to power output, which is measurable through a calibrated bicycle or treadmill. And we can begin to look at the VO2 measured by gas exchange then is ultimately a reflection of aerobic exercise intensity or aerobic fitness. So we have an increasing ATP demand with increasing exercise intensity, and the VO2 that you're able to achieve at your symptom-limited peak or your maximum will therefore represent your maximum ability to drive ATP production aerobically. So the higher your VO2 max, the higher your aerobic fitness is. the higher your rate of oxygen consumption at what we call the ventilatory threshold, or the lactic acid threshold, or the onset of blood lactate accumulation, or the ventilatory threshold, whatever you want to call it, they're basically all the same thing. The higher the VO2 at this break point or this transition between sustainable and non-sustainable exercise, the higher your cardiorespiratory fitness. Okay? So, we get to the point of two things that we can measure. This becomes important for the lab because we can measure VO2, we can measure power output, and therefore we can get an indication or some relationship between the aerobic production of ATP relative to somebody's external power. Right? So, how do we then match ATP supply and demand at the onset of exercise? Going from seated to standing, going from resting to cycling, going from walking to sprinting. How do we match this immediate increase? How do we protect ATP at the site of the muscle, provide ATP at a higher rate to support the higher demands, but without compromising the concentration of ATP at the muscle? Because we know that we need a matching of supply and demand. We know we have three energy systems. Some are faster than others. So what's the interconnectedness and what's the interrelationships between these energy systems is where we're hopefully going to be going here. So if we measure VO2 at the onset of exercise, okay, so we go from rest, and then at this point here, arbitrary time point, in 3, 2, 1, start pedaling on a bicycle. Immediately, the oxygen uptake requirements increase because ATP demands increase immediately. But if you measure what they call the kinetics, the time course of change in VO2 from rest to when the oxygen uptake reaches a steady state, it doesn't happen in a step increase. It takes a little bit of time for the VO2 to respond to that increased demand. There's a sluggishness. Okay? So the VO2 kinetics actually look something like this. You get, this is the, I wish this pointer worked. You have a VO2 demand, which is an immediate step increase, which would be the activation of oxidative phosphorylation. This is going to sound weird in the recording. But you don't have an immediate increase in VO2. It's a little bit sluggish. So if VO2 is then a reflection So if VO2 is then a reflection of ATP demand, or production through oxidative phosphorylation, you would then assume that the ATP response, or the oxidative phosphorylation response, would mirror that VO2 response, and that it would take a little bit of time to achieve this steady state or ATP supply state.
Wouldn't the immediate need for ATP not come from oxidative phosphorylation?
This is exactly where we're going to go. It's a perfect question. So what we're trying to do right now is set the stage to say that it takes some time for oxidative phosphorylation to turn on. Ideally, VO2 demands would go up and you would be able to supply that, but that's not the case.
So just because you're stored ATP thinking?
Yeah, so we'll begin to break that down in terms of that interconnectedness. So here's what we have is what we refer to as the oxygen deficit. Okay, so it's basically that difference between the immediate increase in the demand for oxygen and the amount of time it takes for VO2 to reach a steady state level. Okay, so if your oxygen deficit is very, very high, which is to say a big area, okay? So if you had an oxygen deficit that looked like this and achieved a steady state much later, you have a very big oxygen deficit. What would that say about the time it takes to activate oxidative phosphorylation? Would it be fast or slow? Slow. Now, if our oxygen deficit looked like this, and we had a very, very small area subtended by that demand to supply curve, what would that say about the rate at which oxidative phosphorylation is able to be turned on? It's quicker. So ideally, we would like to have a small oxygen deficit, because it would be an indication that oxidative phosphorylation is able to turn on very, very quickly and start producing energy for muscular contraction more quickly, and to your point, not rely so heavily on other forms of ATP liberation. Stored ATP breakdown, phosphocreatine, hydrolysis, and things like that. So, again, coming back to what we showed previously, the fact that VO2 is directly related to the rate at which ATP is produced through oxidative phosphorylation, shown in the previous slide. And the fact that VO2 does not increase to steady state levels at the onset of exercise certainly suggests that, to your point, which is a very good one, anaerobic pathways must contribute to the overall production of ATP in the rest exercise transition. Because we know we have to match ATP supply to demand. But we've got 3 different ways to do it. We can't do it all through oxidative phosphorylation at the immediate rest exercise transition. And that it also suggests that there's a progressive turning on of this oxidative phosphorylation. So there's a bit of an inertia that needs to be overcome. Like that old Cutlass Supreme. It takes a little while. The engine will get going, needs to get warmed up, but once it gets warmed up, it can supply the energy demands that are required for that particular task. Yeah.
Let's go back to this. Is Shirley safe for oxidative phosphorylation to stop?
Yeah, so in that case, you get what's called an oxygen, the oxygen debt, and that can be measured as well. And that's basically an indication of how quickly the energy stores are reestablished back in the muscle. So some people use the oxygen debt as a measure of basically muscular health as well, and oxidative capacity as well. So yeah, you can measure what they kind of call the off-transient response.
But then also be measured in minutes, or would it be?
Yeah, again, it's a rate. It's a time course of change in oxygen, in this case a decline back to some resting level. So instead of this being an upward curve, shifting curve, it would be a downward shifting curve. So, coming back to this idea of ATP demand, to your point, in order to match ATP supply to demand, we have to have ATP being supplied through phosphocreatine hydrolysis and anaerobic glycolysis in order to make sure that ATP at the site of the muscle is delivered and that there's not a decrease in ATP concentrations, because as we talked about, it's a regulated variable. So, but the relative contribution of these energy pathways will decrease as the relative contribution or as oxidative phosphorylation begins to turn on. Okay? So you can appreciate then that the oxygen deficit is an indication of how quickly oxidative phosphorylation turns on, and the corollary of that is how much we rely on the anaerobic or non-oxidative pathways to support ATP. We have a big oxygen deficit, we have a big metabolic disturbance in the rest exercise transition, which could set the stage for earlier fatigue, right? You get a big rush of inorganic phosphate and hydrogen ion just by trying to support a muscular demand in the rest exercise transition. That muscle is a little bit more pooched than it would be. It's basically starting from a worse starting position than it would otherwise be. So trying to have a small oxygen deficit as an indication of activation of oxidative phosphorylation would be, a goal of, say, training, for example. So, since VO2 is directly related to the rate at which ATP is produced aerobically, it's not surprising that the kinetic or time-dependent response for oxygen uptake measured at the mouth using a metabolic cart mirrors that of oxidative phosphorylation. Again, what you should be doing now is linking these particular assumptions that I outlined in those 4 graphs on the previous slide. Now, if you looked at the time course of changes in phosphocreatine and inorganic phosphate in this rest to exercise transition, what you would see is that they would go from their steady state resting levels, and you would see that phosphocreatine levels would degrade, reflecting this phosphocreatine hydrolysis, and as a result, inorganic phosphate with the breakdown of ATP, stored ATP, you would see that inorganic phosphate levels would rise. And again, they follow a similar kinetic or time-dependent time course. So because ATP synthesis at the start of exercise relies heavily on phosphocreatine hydrolysis, and because it takes a while for oxidative phosphorylation to turn on, it's not surprising, again, that phosphocreatine levels decrease and inorganic phosphate levels increase progressively to a new steady state, but along a similar kinetic or time course as the activation of oxidative phosphorylation. Okay? So the question then becomes, what's responsible for this metabolic inertia? Why would the system be designed like this? Why wouldn't we just be able to jack up oxidative phosphorylation immediately, have no oxygen deficit, and have no muscular disturbance? Well, there needs to be apparently some signaling between these different energy pathways. So, as a summary, basically, the energy needed for exercise is not provided by simply turning on a single bioenergetic pathway, but rather through progressive, coordinated, and interconnected, and interdependence, a lot of words, activation of all three ATP-producing pathways. So you can appreciate, if you look at the different systems, the ATP, PCR, glycolysis, and aerobic systems, is that you have a very, early on in exercise, you have a very high contribution from the ATP-CR system. As it's basically decreasing in its contribution, you're getting an increasing relative contribution of glycolysis with a progressive and slower activation of oxidative phosphorylation. And again, you should be able to kind of appreciate the time course of these changes in relation to the VO2 and kinetics that I showed you on the previous slide. So, some kind of basic concepts. In terms of the control of bioenergetics, what determines flux through an enzymatic reaction? So we have substrates, products, and the enzymes. Okay? So, to increase flux, which is basically just not like, Back to the Future flux capacitor. This basic flux is movement from one position to another. To increase flux through this enzymatic reaction, we can increase the number of enzymes. So, one of the things that exercise training does is increase the number of mitochondria in a muscle. If you have more mitochondria, it means you have more mitochondrial enzymes. So it's basically, when they talk about mitochondrial density, part of what contributes to muscular endurance is that when you have a greater mitochondrial density per muscle fiber area, you have obviously more powerhouses of the cells, but you have more enzymes concentrated within that cross-sectional area of the muscle. So greater mitochondrial density means greater mitochondrial enzymes. So for any given substrate to product ratio, more enzymes should mean more flux through this enzymatic reaction, one of which is oxidative phosphorylation. Or you can increase the activity of enzymes. So you can have these enzymes that are basically latent or not particularly active, but need a trigger. So regardless of their concentration, they need to be activated to drive these enzymatic reactions more quickly. So turning on these existing enzymes. Now if you think about it, the breakdown of ATP and the accumulation of ADP, inorganic phosphate, lactate, hydrogen ion, for example, would all be great signals to turn on these rate-limiting enzymes, the isocitrate dehydrogenase, CK, phosphofructokinase, right? Because it communicates the state of the muscle. ATP demands go up, ATP at the site of the muscle breaks down, ADP goes up immediately, calcium is released from the sarcoplasmic reticulum and immediately on the contraction, inorganic phosphate goes up, muscle's like, shit, I gotta do something. How can I increase the flux through this, and how can I try to minimize this oxygen deficit and increase ATP provision through oxidative phosphorylation? As an example, activate the existing enzymes, turn them on. So some of these byproducts, which actually are coming from the ATP PCR system and the activation of glycolysis, are the factors that will turn on these Krebs cycle enzymes and these mitochondrial enzymes and increase their activity to try to minimize that oxygen deficit and try to match ATP supply demand as much as possible through an oxidative means. You can also, of course, increase the substrate to product ratio. So if you were to, as I'll show you some examples, if you were to increase the amount of oxygen, you can drive ATP and through the flux through that enzymatic reaction more quickly. So the implications for this intracellular metabolic milieu at a given rate of flux is that the same flux through an enzymatic reaction can be achieved with a lower substrate to product ratio if there's more enzymes or the enzymes are more active. Ladies, you three kind of keep looking at the same monitor. I don't know, and it's like some giggling and things. I don't know what you're doing, but I don't know how entertaining this is. I find it entertaining, but I don't see there's like 60 other people that aren't kind of looking at the monitor and smiling.
You're up on the same window.
Okay. So, in general, if there is ample substrate, for example, if there's enough oxygen in the cell, as one example, an increase in the number of enzymes or the activity of existing enzymes will increase the rate of flux through an enzymatic reaction. We talked about that. So, chronic exercise training, as an example, serves to increase the number of mitochondrial enzymes by increasing the number of mitochondria, which certainly accomplishes number one. But the question then becomes, what bioenergetic factors are responsible for number two, stimulating the existing enzymes, regardless of their concentration? So, as I mentioned before, ATP itself is actually an inhibitor of enzyme activity. If you have, for example, if you have an excess amount of ATP in the cell, it's kind of indicating that we don't have much energy requirements, right? And so, as a result, you're probably going to decrease enzyme activity because you don't need to drive that enzymatic reaction particularly aggressively. However, increases in ADP, inorganic phosphate, and calcium, which is released from the sarcoplasmic reticulum into the cell, stimulate enzyme activity. And again, this makes sense. You want to have these signaling molecules that will activate these enzymes in relation to the energy state or the energy demands of the muscle. So again, this makes sense.